Thank you so much for being here for the final session. So I'm going to talk to you about more manipulations of the focal point plasticity plugin, and we will be using it in the context of actually epithelium tissue folding, something that was talked about a little bit in the last lightning talk round. And so hopefully this will give you a better idea of what the capabilities of the FPP plugin is and how you can manipulate the properties both from using the XML files and the Python steppable files. Okay, so just to start us off, so the topics that I'll be covering will mostly be one part of it you have already seen with Julio yesterday. So we will be using that as our base simulation to carry on and look at some more FPP properties. So our first goal would be just creating our basic epithelium and making sure we can define some FPP links in between these compartmentalized cells of the epithelium. Then we're going to manipulate these properties to actually get a fold in the tissue layer. And we'll see how we can do that by using the steppable file in Compusol 3D. And then the last three modules, 7.3.3, 7.3.4, and 7.3.5, they are a little bit involved coding wise, but the main goal from the last part is sort of how we can manipulate these properties both in space and time, so that you can get, for example, in our case, we'll be looking at an example of breaking the whole tissue into discrete units. But you can use these properties if you know how to sort of like code for this in the steppable. You can use it for whatever purpose you see fit. Okay, so I'll start off with some basic motivation for this. So our goal on why we are actually even interested in tissue folding comes from the following. So development has this wide array of examples in which tissues greatly change their shape to perform specific functions and objectives. So for example, on the right, you have examples of gastrulation from fruit flies or C. elegans, where during the process you have these tissue folds that appear, and this actually helps specifying the germ band layers. In this case, it's in the fruit fly case, it's helping bring in the epithelial surface, and which will like downstream regulate many other steps. But then this process of like being able to bend the tissue or fold the tissue becomes important. And similarly, in the context of neural tube development, you have sort of like a flat sheet, and then you're able to like bring together different sides of the tube to have these like a folded tube morphology. So all of these examples of tissue folding, they basically rely actually at the cellular level, because in these cases, the cells are in fact connected by these cytoskeleton elements, which are able to exert forces and bring cells together. So for example, the leftmost example that I'm showing here is a case in which you have an epithelium, and because of specific cytoskeleton elements, in this case, it might be an example of actin myosin contraction, you're able to bring the apical surface closer together. And as you're able to do it, you can see that a rounded, bendy shape emerges from the tissue. There are other examples that could be coupled in it. For example, the middle case shows you a situation where there might be a dead cell in between. So if you have forces that are exerted on either side of the cell, then that cell is sort of like extruded outward. And you might have shape changes at the level of the neighboring cells as well. So all of these examples in which you have changes at the cellular level, morphological changes at the cellular level, which can then impact the tissue structure as a whole and is able to generate these like complex shapes that we see. So our goal sort of for today is we'll have like a very basic epithelium, and we're going to use Julio's definition from module 6.1 to divide this into 3 different compartmentalized cells. We'll call them apical, basal, and everything in between. We'll call it a lateral cell compartment. And our goal will be to sort of being able to exert specific forces on the apical side to get this rounded shape. And so the code for this module can be found in 7_3_1. So we have 2 choices now. Because Julio has gone over this example, you are free to look at the objectives for here, and you could do this on your own, or I will be actually just repeating through some of these steps because we'll be adding the focal point plasticity as we go along as well. So if you would like to go along with me, feel free to do so, or you can also just download yesterday's simulation, this simulation, or you can build your own in the meantime, whichever one you'd prefer. Okay, so what are our objectives so far? So we're going to build this epithelial tissue layer. We're going to have 4 cell types, apical, lateral, basal, and ACM. And we will be adding this plugin, additional plugin, the focal point plasticity. We will mostly be dealing with the uniform initializer to build us that nice layer, which Julia showed yesterday. And I'm sure by now you're also familiar that there are properties like cell field that you can also call in the steppable file to actually create or put cells in a simulation as you see fit. But after we have done this for the simulation, we will modify our FPP properties so that our links are only on the apical side. We'll be adjusting some of our internal contact energy parameters. And then we are finally going to make sure that in the steppable file, we're able to manipulate the apical side properties. So this will be part of the 7_3_2, but just setting up the simulation and putting basic links on the apical side is under 7_3_1, if that's what you want to pick. Okay. For those who are going to follow along with me, these are the steps we're going to take. So you can open to edit and set up some simulation features. So in our case, these are the dimensions that I have chosen. For the x-axis, you can put 350. On the y-axis, you can put 100. And then the x-axis boundary conditions you can set to periodic. The other properties, you can still set it to a square lattice. Average membrane fluctuations could be at 10. Number of Monte Carlo steps, all other parameters good. And then you can hit next or continue, whichever version of TwoEdit you're using. Yeah, so these are the 3 basic things that we're changing from our standard simulation. On the next page, what we're going to do is we're going to add 4 different cell compartments, which is apical, lateral, basal, and ECM. And don't forget to hit add as you write down each cell type name. And once you have done that, we're going to ignore things in the chemical field window, but we are going to make sure we have these additional plugin selected, which is the focal point plasticity. So make sure you have contact selected, you have contact internal selected, you have focal point plasticity selected, volume plex, and then you can put in center of mass and cell neighbors, which are automatically selected, I believe. So with this, I think we have all the components of our simulation here, and we will be going on step by step to see how we can use the uniform initializer property to use the simulation. Okay, so for those who are following with me, so if you have specified the dimensions of the box correctly and the uploaded the plugins correctly, you should be able to hit step in the simulation, and you should be able to see this output. I'm going to wait for a few seconds to confirm that this is what you see. And so the question is, how do we know to use volume plex rather than volume local plex? So it's so volume local plex will allow to change the will allow us to change volume properties locally in the FPP in a steppables file, if I'm not mistaken, and anybody can correct me, but I believe that that was the difference. Like if you are planning on incorporating incorporating other volume changes in the steppable file, you can use the volume local plex plugin. All right, so so if people have made it to the step, if anybody wants to share a screenshot, I'm gonna wait for like a couple more seconds and move on. But I hope that people have the basic simulation or have the complete 7_3_1 downloaded. So right now, what we have is when you give these different cell types to uniform initializer, depending on what the width is, it's going to randomly fill the chosen area, like the minimum and the maximum, and with these cell types with this given width, and it's doing this randomly. Awesome. Thank you, Vikram. So this is sort of like the base simulation that we do want to modify. So as you saw yesterday with Julia's case, what we're going to try to do is use these box minimum values, box maximum values to manipulate to get a nice layer where we have ECM at the bottom. Then we are going to put in a basal compartment, a lateral compartment, and an apical compartment. So basically, we're trying to follow the structure of this type of cell. From epithelial biology, what's usually the case is that the basal surface of the cell is usually on a substrate, and which is like made up of these extracellular matrix proteins. And there are like these connections between the basal side, which are called integrins. And so the basal surface is like likes or prefers to be next to the ECM layer. And then on the other side, facing the, let's say, the outer surface of this layer, that's where the epithelial body usually lies. And there could be other structures like microvilli that allows it to sense the outside environment. And along the body of the cell, you have these bunch of connections, which could be made of the cytoskeletal components that I'm talking about. And keep in mind that this is like a very dense cytoskeletal network that connects, goes through like the cell bodies, and that's how it's able to like bear these loads to help the cells change shape. So that's the basic idea between trying to modify the properties of the uniform initializer so that we could build the structure where we have ECM at the bottom, basal surface next, then lateral, which we are just calling the sides, the additional side of the cell. And then we will have this apical compartment. So our first step that we're going to follow is sort of modifying this property. So in the uniform initializer, where you had the values for X, box min X and box max X, or Y, min Y and max Y, let's change the values for X to 0 to 350, for Y to 0 to 35, and then you can set the width of this to 35 and make sure that you have the only cell type that you have is the ECM. So right now what we've done is just laid down the foundation of the ECM layer. Okay, hope that makes sense. And the next thing we're going to do is, so right now we have only specified the ECM layer by using these minimums and maximums. And then as we're going to need our additional cell types, we're going to go to this step where we're just going to copy this entire block. If you just copy this entire region to region block and then add another one, now what you're going to do is in the Y minimum and maximum, you're going to put 35 and 42. So you're basically specifying the height along the Y axis for another cell type, which we're going to call basal. And you can set the width of this to 7 as well. And that should give you a cell layer where you have ECM and then you have basal. And I'm going to repeat this 2 more times before checking in with you. So next I'm going to add the lateral cell type, and then I'm going to add the apical, and I'm going to add the apical cell type as well. So I'm going to pause here for a second, check in with you if you have been able to get the 3 basic layers down along with the ECM layer.
Are you building these layers specific to a publication that you flashed earlier, or are the values for Y a little arbitrary for a demo?
Oh, right. I mean, you can put any values of Y here. Yeah, these were depending on how many cells you want in your simulation, these values are arbitrary, yeah.
We might have reviewed it, but you need at some point to review how you take something from a publication data and make sure it's matched in the simulation.
Right, right. So usually, I mean, and there are a lot of different ways to do it. So you could measure some features of the cell. For example, if you have experimental data and you're able to match and you're able to measure what the size of a cell looks like. And then you can make a choice here to say that corresponding to this in my simulation that I'm trying to produce, there are these structures which are about like 100 microns that I see. And you could backscale from there to say that if these are the structure that I need to build of like 100 microns in the simulation, how many cells do I need to put to achieve that structure, and how many structures like that do I want? And then you could say that, oh yeah, 10 microns is equivalent to say 5 pixels in my simulation. And that and you can make that choice. There are also constraints in CompuCell. For example, if you have 2 tiny cells, then it's not going to be a very good calculation from CompuCell wise, and the cells may like fragment out too easily. So there has to be a minimum limit that you choose for your cell, and then there is a scale that you pick from just measuring your experimental parameters and sort of trying to estimate how many pixels could correspond. In this case, this basic simulation does not correspond to anything in particular, but for example, if I was mapping any structure or feature, then I would pay more attention to how many pixels I'm choosing.
Thanks for that. That was really helpful.
Yeah, and I think Sharon's making a very good point about, what's not arbitrary is the aspect ratio of the compartmental cell. And so if that's the feature that you want to map, and you're like, this is the ultimate size that I want to reach, and so those are kind of the features that you try to match from your experimental images. Let me quickly check. Yes. So the Twitted is interesting because so I was using 4.6.0, and if you just hit CompuCell from Twitted, it was crashing on mine. But if you separately open CompuCell and then just load the simulation, then it seems to work. So I don't know if it's a 4.6.0 version on using a Linux machine versus if it's anything else, but I will definitely make sure to let people know. But yeah, if you open it from CompuCell and then hit run or step, then it should work. Okay, all right. So moving on. So awesome. So you guys have this basic tissue layers and we have the ECM. So our next step is sort of going to be actually visualizing what the FPP links to. Before that, if you're planning on running the simulation, let's do one quick thing where if you go to your volume plugin, make sure to set the target volume of this, make sure to set the target volume to 1225 to just match what the correct ECM volume is, because we have changed the width from 7 to 35 here. So after doing that, if you run the simulation and make sure you have FPP links selected under visualization, you should be able to see things like these white lines appearing between compartments. And because we have periodic conditions on, you might also see this like last compartment cell trying to connect back with the first one. So what we have done so far is in our FPP plugin properties, which we're going to modify next, we have all of these compartment links set up because Compusel loads them automatically. And so once you have like set up your cells, focal point plasticity is automatically trying to assign links between all of these compartments. And those are the lines you're seeing. Okay. So if we have this sort of simulation so far, next our goal is to sort of be more concrete with our FPP links, because remember, our goal is still the same, that we want to specifically fold a tissue along the apical side. And if we want to put additional forces, we really don't need most of these other links between compartments. And so we're going to get rid of them. By the way, we are only exploring the condition where we are sort of trying to like fold the tissue. But if you wanted to explore other properties like elongation of cells, one possible way, instead of using the elongation module, is also putting links in between the compartments. So if you put a long link between your basal and apical side, and you might have to balance additional links on the apical to lateral and lateral to basal. And then if you sort of like start increasing your FPP target distances or target properties, you'll be able to also elongate the cell. And depending on how fast you elongate between the apical and basal versus apical to lateral or lateral to basal, you might also be able to get different cell shapes and bendy cells, in fact. So that's also something, if you are interested in exploring focal point plasticity, are helpful. You can't see the FPP links after selecting in visualization. Could you check if the color is selected correctly? Don't know why that would not be. So there should be, if you go to the, yes, thank you, that'd be great if somebody can help you. If it doesn't work, in the meantime, you can also download 7_3_1 and see if the base simulation at least have the FPP links. Thank you. Okay, so right. So as I was saying, our goal is the following, that we are trying to sort of fold the apical region in this case. And so our links need to be specifically on the apical side and not on any other surface. And so this is precisely what we're going to do. And just as a reminder from yesterday, what the focal point plasticity plugin was doing. So what it does is, given any two cell types, in this case, we just want it, in this case, we just want it between the apical and the apical compartments. These are the cell types you can choose. The other parameters are lambda, which basically dictates how strongly you want to pull, or you want the cells to be pulled to each other using the focal point plasticity plugin. There's this component of activation energy that sets up that sets up an additional term to make the building of this link more favorable. There is the target distance that sets out how what is the target distance between the compartments you want to achieve. There's also this maximum distance beyond which the focal point link will break. It will no longer try to form beyond that link. And you can select the maximum number of junctions. Basically, it dictates how many how many links from each cell it's going to form. So what we're going to do is going to sort of manipulate some of these features. And so now, if you go back to go to your XML file under focal point plasticity, we're going to do a couple of things. We're going to uncomment this local tag. And then we're going to delete almost everything. We don't need links between any other cell compartment, so we're going to remove all of them. The only link we are going to keep is between the only link we're going to keep is between the apical compartments, like this. So there's a question, if there's a maximum of one link per cell, are they then asymmetrical? That is, the link belongs to one cell, but not the other. That's correct. That's exactly correct. So if there's one link, I believe the simulation tries to set it up randomly, so you don't know which, if there are two cell pairs on either side, you don't know which cell pair got it, but it will try to just assign one junction on either side. So as you can see in this simulation right now, the maximum number of junctions has been set to 1. So it's not possible for all of these cells to have the link in between them. There are some cells which I do not know why did not get, sorry. There are some cells which, for example, this pair did not get a link. So we can increase this number to 2, and then we should see links between all the pairs. Shouldn't the interactions be symmetrical? So all the properties are symmetrical, except that if you put a neighbor order of 1, and if there are multiple neighbors, it doesn't have the option of forming more than one link, and that's the problem. But yes, all the focal point plasticity are like pairwise. So when you have two cells, you're measuring the properties between that cell pair. So the lambda, the target distance, is for that cell pair. All right, so the question is, why the local specification? We have uncommented the local tag because we will be making some changes in the steppable file as well. And so if we want to modify properties in the local file, we need to uncomment this tag, or any changes we make in the steppable file will not be recognized. Okay, so I hope everybody has the simulation right now. So all we have done is removed everything else for the focal point plasticity. We have set up some things for the apical compartments, but in the next step, you will see what we're going to do is we're going to remove most of these features, because we want to be able to manipulate this in the steppable file. So after you have removed all the other cell tags, if you want to just go to the apical tag that now remains, and you can set the lambda, the target distance, the maximum distance, and the maximum number of junctions to 0, and we'll modify all of these properties in the steppable. I believe in earlier versions of CompuCell, this activation energy term could only be manipulated from the XML, but now you have the option of manipulating activation energy through the steppable file as well. So the question is, what is activation energy? So this additionally adds a favorable energy value to your Hamiltonian term, to your energy function, to make creating this link a little bit more favorable. Can you explain max distance again? After this, links will break in the context of the default value, right? So the maximum distance allows you to set a limit beyond which, even if you have all the other properties set up, for example, you're saying that you have a bunch of cells, and if the cell form links between all cells of this kind. If you set this maximum distance tag along with it and you have a value, any cells which are separated beyond that maximum distance will not form a link. So if you had, for example, you wanted to make a specific zone around which you only wanted to make links and distance was your criteria, that beyond that distance you did not want links to form, you can use maximum distance.
So then in the context here where you set it to 0,
Mhm.
there's no wiggle room, no links will be formed outside of
Right.
any
So here,
separation.
yes. So
I would think
right.
that would, I guess I would assume that would be the case. I'm surprised that there is wiggle room at all. I would think that links would not be possible if there was any distance between the two objects.
Sorry, could you say the last part again?
I guess I would think that there would be no links would be allowed if there was any distance at all, I guess. I would think that
Oh,
links would break if there was any separation.
I see. So you're thinking that, no, but that's a good thing about focal point plasticity. You can form, for example, suppose you started forming links between two objects which were originally touching each other, where your, neighbor order parameters could come into play. Now, suppose for some reason these cells started moving around, the link between them would still remain until a parameter like the maximum distance could come into play. So it's not that the cells have to be touching each other all the time. If you have set it up in a situation where they were touching each other and they actually later on moved apart, the links would still stay between them and you'll be able to see it. Right, and James was adding on to it that links could represent filopodia or just long-range forces, right? So it's not just between, connected epithelium that we're talking about, but for, for cells that could be, migrating through a substrate that it's sort of like anchoring and using these forces to like move along the substrate, you can use focal point plasticity links to sort of sort of like anchor to a separate cell type that's not directly in contact with it as well. So yeah, links do not have to be restricted to just touching cell types. Okay, so okay, so then what we're going to do now that we have modified these focal point plasticity parameters, we are going to do our final step where we're going to make sure our contact energies are in the right zone. So if you remember this from yesterday, the rule that we're following for our epithelium is that we wanted some sort of favorability between components of the cell type so that they did not like sort of hate each other too much, but we did not want it to be too low that they started like mixing, the compartments kept breaking and mixing among each other. So we wanted to set the intercompartmental contact energies to be on the lower side, and within the same compartment you can the contact energy does not need to be specified and you can set it to 0. So we're doing this in these steps over here. So if you look at the contact energies from 7_3_1, this is the hierarchy that I followed, where between the same cell, the intercompartmental contact energies are set to low. And now we need to think what to do for the external contact energies. So if you remember again, our goal is that the ECM is at the bottom and the basal side is close to the ECM side. So we definitely want that the ECM to basal interactions are on the lower side. But we want that the compartments of separate cells are also sort of like they prefer each other rather than other compartments. For example, apical compartments of two neighboring cells prefer each other versus apical compartment of one cell to the lateral compartment of other cell, so that the epithelium structure is maintained. So we'll be following that hierarchy as well. Additionally, that the apical compartments prefer to be next to the medium or so. So what you can see is that on the medium to apical side, you have like, again, lowish contact energies, similar for basal to ECM. And then between the same type of compartment between neighboring cells, you have mostly like lowish values, lowish values, lowish values. And then you don't want apical, obviously, combining with the basal of the neighboring cells. So that's why the contact energies are set to sort of like on the highish sides. And you can follow roughly, the values don't have to be exact, but you can follow roughly the same hierarchy to sort of make sure that the same compartments of the neighboring cells line up next to each other. And so the values that I have picked are following. And you can either pick these values, or you can make up your own hierarchy as well. And after this, if you have build this range, then you should be able to see that the apical compartments are next to each other, the basal compartments are next to each other, and the lateral compartments are next to each other. The other thing that we have done is the ECM contact energies have been set to 0. So they are just mixing around and forming like a giant ECM blob rather than having any compartmental boundaries. So I'm just pausing here for like a couple of seconds to make sure you're on the same page and you have the simulation. So far what we have done is removed all of our focal point plasticity parameters, keeping only the apical and apical ones. And then we have set up most of these parameters to 0. And our goal is that we'll be modifying most of these in the steppable file. That's why we have the local tag under the focal point plasticity plugin on. And then we will be modifying most of our properties there. Can you show again the contact internal numbers? Absolutely. So Sharon's question is this just an arbitrary way to get the cells to behave, or does it model actual biology? Do basal ends of cells actually recognize and have affinity for each other experimentally? So yeah, somebody better equipped with epithelial biology can give you more details, but yes, there are, for example, like specific level of these cytoskeleton elements that is present along from the apical basal side. For example, these integrins are only present on the basal side that helps it to connect to the components of the ECM. So that's why we are modeling contact energies for the basal favoring the ECM. The fact that these compartments are lining up also comes from biology, because you have structures of like intermediate filament, you have apical fibers, which are present also, like the actin myosin fibers are present more on the apical side of the cells. The intermediate filaments might be present more on the lateral side of cells. So the hierarchy that we're following for the basal, lateral to apical is sort of building on the hierarchy of the of all the cytoskeleton elements that are present, and the body of the epithelium that they adopt. And like I was saying, the apical side is in contact with the outside surface because there might be additional like processes like microvilli for it to like actually sense the outside surface as well. So that's why we want the apical side, for our case, to face away from the ECM side. Okay. So hopefully we're good with the internal contact energies. Going to spend maybe a couple more seconds on the external contact energies, in case anybody still is struggling with this. I'm going to move on to our steppable file. So now what we're going to do, I'm sorry. Now what we're going to do is modify our steppable file. So I'm sure you know how to find it by now. If you go to your Twedit window, then you can click on the steppable file, and this should bring you to this window where you can see a bunch of different functions, the initialization step, the starting steps, the step Monte Carlo step function, and the finish function. I'm sure you have seen this already. So most of our we're going to use for our purposes both the start and the step functions to incorporate some code. So in the start function, we're basically going to, before any of the simulation starts, we're going to reassign some of our cell properties. And in the step function is where we are going to change some of our focal point plasticity parameters. So there's a question in the chat, is there a way to model ECM rupture? I am actually not familiar with the phenomenon. If you describe it, I might have better thoughts on it, or if James has any ideas on that.
So now or later?
We can talk later if that works for you.
Yeah, okay. Thank you.
All right. So what we're going to do is let's focus on the start function for now. And we are going to modify a few things in the start function. So if you have not seen this function before, which is self.celllist by type, we are going to be using this to sort of look at all cells of a specific type. So our loops are the following. We're going to first loop over all of our cell types, which is in this case the apical. And then once we have found out all the cells of the apical type, we are going to find the lateral and the basal cells, which we think should belong to the same compartmental cell. And we're going to make sure that these compartments know that they actually belong to our giant cell cluster. And why we're doing it is the following. So right now, all of our cells that we have built, all of the cell types like apical, basal, lateral that we have built, they are individual components. They do not know that they are actually part of a larger cell. And this is where the things you've learned about the compartmental cell comes into picture. So until we sort of like reassign the cluster IDs, they do not know that they belong to the bigger cell cluster, and their properties the properties that you want to be modified or the properties that you want them to behave as part of a giant cluster won't be completed till you have done this. So what we are doing is we're looping over all these cells under the apical. And then what we're doing is we're writing out another loop. So now we're trying to find out all the lateral cells. And the way that we know that two cells will belong to the same cluster is by using the following condition. We're just comparing the center of masses, because if you remember from our simulation, we were just stacking up all of our compartments along Y, which means that if we want our cell to be part of the same compartment, they have the same center of mass in the X direction. So if this condition is met, that these two lateral cell has the same center of mass as the apical cell, we can safely say that they are part of the same cluster. So that's why we're saying reassign the cluster ID of your lateral cell to the same cluster ID as the apical cell. And then you can do the same for the basal cells in another loop. Does this make sense? So right now we're trying to assign all of these cell types to sort of be part of the same cell. And then we're doing this by assigning the cluster IDs of these subgroups to be the same of the apical cell in this case. You can also choose maybe the lateral cell, and you could have assigned both the apical and basal to the lateral, or you could have a bigger loop for the basal cell, and you could have assigned the lateral and apical to the basal. whichever way you prefer. Here I have looped over the apical. Hopefully this makes sense. Okay, so then so after that step, we're going to do one more thing. So in this part, we have only specified the compartmentalized cells that they belong to the same cluster, but we haven't done anything with our focal point plasticity plugin, which we just removed. So we're going to do one more step where we're going to loop over our apical cell again, because we're only forming links between apical cells. And then we're going to use this plugin of visiting all our cell neighbors, which can be found under CC3D Python, visit cell neighbors. And then we're going to say that if you are a neighbor of this apical cell, and if your neighbor type is apical, then we're going to set up a link, which is going to be in this step, and this is the complete code. So we're going to, for the link setting part, we're going to CC3D Python, focal point plasticity, link management, and then we're going to create this FPP link between these properties. Okay, so I think there were a couple of steps. I'm just going to sum it up again. So in the first step, we went to the start function, and we were trying to make sure our cells know that they are part of the same cluster, that it's actually a compartmental cell calculation that we're doing so that the contact internal energies can, kick off into play here. And the how we did that was we reassigned the cluster IDs of these compartmentalized cells to one cell type, which in our case was the apical. And then what we did was we now wanted to form our FPP link. So we went to CC3D Python. We first visited all the neighbors of that particular apical cell. We said that once a neighbor has been identified, let's try to form a link between that cell and its neighbor. And I'll talk about the parameters in a second, but I just wanted to touch base if you were able to get to the simulation or you were able to have this. So there's a question on what is activation energy. Yes, like I was saying, activation energy is this additional favorable term that you can add to your energy function to make creation of the link more favorable. And in this case, I should point out, because we have overridden all of the focal point plasticities, now we are looping over the neighbors, and that gives us more freedom on choosing, how we want to form our links. In this case, we have chosen that if any apical compartment is present between, there is another apical compartment in vicinity as a neighbor, you can form that link. And that's why now you can see this whole connected sort of line connecting all of the compartments. Right, that's a good point. Does it make one link or two? One link will be visited twice by the left and by the right side, right? So it will only make one link, but you're right. I think an additional check, you can put an additional check in this loop if you wanted to say that if that cell pair has already been visited, you don't need to visit it again. Yeah, but there will only be one link that will connect the two centers of masses. Yeah, does anybody want to share any pictures or any other issues so far? So at this stage, we should have compartmentalized cells, and then we should have the focal point plasticity links in between the cells. Oh, perfect. Thank you. That looks great. So we have links in between our apical sites now. That's great. Okay, so now you have other things that I haven't talked about are what these parameters look like. So once you have created this new FPP link, which is between the cell and the neighbor, you can additionally specify the same features that you saw in the XML file. For example, the first parameter is your lambda distance. So basically how strongly the link is going to pull on the center of masses between the two cell types. The 7 is basically the target distance that you want to achieve in between the two compartments. Right now, the assumption is if you have a cell which is a width of 7 and you're connecting the center of masses, that's going to be the typical distance. And you will see in a moment how we can like change this parameter to achieve different tissue shapes. And this is the parameter for the maximum distance, which is 200. So if any cells got separated out beyond this, the links would have broken. So next, our goal is going to be the following. Now we want to use the FPP properties to be able to bend the epithelium. So I have a question for you guys. So far we have like links in between the cells. Suppose we want to achieve this particular shape, what would we do next? And what are some of the things we could change in our simulation to achieve this? Decrease target distance? Yeah, absolutely. That's one possibility. So would you decrease target distance everywhere, or would you need to do something along with it locally? Absolutely. So one of the things you could do here is like decrease the target distance locally, because that would apply a more specific force in a specific region and increasing the forces that would allow you to have this bend shape. So that's exactly what we're going to do. And what we're going to do is now we're going to use the step function, and we are going to use the exact concept of applying local forces. And we're going to specify our, we can use the center of mass again to specify sort of a local distance. In this case, maybe we can, I have picked out 125 and 225 as between the minimum X and the maximum X. And you can definitely pick your own. So what we're saying is, if you are a cell belonging to cell type apical, and if your center of mass lies between these two positions, then you can collect the link properties again. And we're going to do this by going to CC3D Python, focal point plasticity, visiting all links attached to a cell. So if you fulfill this criteria, let's get the links that have formed in this region. And we are going to change, in this case, the lambda distance, which is the force with which you were applying. And we're also going to change our target distance. So changing the lambda distance is just an additional measure to making sure it's able to achieve this target distance. So we're basically increasing the force by making sure the lambda distance is set to a higher value in between those cells. The other feature that I want to quickly point out is that I have chosen this at a particular time. So basically, I have let the simulation relax up till a certain point. In this case, it's 500 Monte Carlo steps. And then we're doing our tissue folding example at that particular time step and in between those particular regions. So I hope people have this right now. So again, two things, we're picking up a particular time step using the step function, and then we are choosing a particular region between which we are trying to modify our FPP link properties. Sorry, there was a question?
Yes, I have a question. So, if I'm not wrong, the lambda distance registers kind of like the rigidity between the two center of masses between the two cells. So, what if I change just the lambda distance and keep the target distance unchanged?
Yeah, you can absolutely do that. I think what's going to happen is you will definitely still see some shape changes at that level. You might not see as invaginated a surface if you don't change the target energy. You will still see that the cells have like clumped together. Cells have clumped together in that region, but you might not be able to see like a more elongated shape because the target distance between the compartments might not have changed that much. Usually what happens is the apical compartment has a fixed number of pixels and width that it's trying to maintain. And so when you change that target distance, what it's going to try to do is sort of like push the pixels in a more elongated fashion so that it sort of is able to achieve the target distance. So the combination of both of them leads to a more leads to a more constricted shape.
And like higher lambda distance registers kind of like low uh, flexural rigidity, if I'm correct,
Yeah,
between the
right. So yes, the lambda distance is proportional to sort of the force that you're able to apply is proportional to the lambda distance. So the greater lambda distance is sort of the bigger force that you're pulling on between the cells. Yeah, lambda will be equivalent to the Young's modulus. That's
Got it.
correct.
Thank you.
All right, so perfect. There are a couple of simulations here already, so you can sort of see the invagination shape already in the tissue. Right. So this is an example of how using particular focal point plasticity parameters using the steppable file, you can like apply things locally based on your simulation. If you needed something that changes as a function of time or space, you can use the step function in a steppable file to incorporate these plugins at a particular point in space and time. So this is great. We could try a couple more things. It's along the same lines as you were saying. You could try changing the lambda distances, and you could try changing the target distances, and you could see how the tissue shape actually changes. And I don't know if that's something people can try. The lambda distance value that we chose was 70. You can set it to 30 if you would like, and you could also set it to 120 or 150 if you'd like. And you could also try setting it to a very large value and see what happens. So your cells are together because of a combination of these internal contact energies and the right mixture of these lambda distances. If you had like an exorbitant focal point parameter that you were trying to achieve, you could in fact like tear up your cells as it tries to like satisfy this constraint. So there are always like values and limits that you can check with these parameters. And I guess you have already answered this question of instead of picking up a small region, if you had, lambda distance for all links uniformly, you would not have been able to achieve this feature because we need this application of specific local forces to have these asymmetry in the tissue that's creating this folded shape. Okay, does that sound good so far? Are there any questions
I have a question, maybe not related to the actual exercise, but if I put to visualize cell glyphs, there are some weird things that happen. What's actually a cell glyph?
What's a glyph? Could you spell that? Sorry.
It's in the visualization and cell glyphs. I don't know.
Oh, okay.
I would recommend finishing this module and talking about that in the discussion afterwards.
All right, thank you.
That sounds good, and maybe one of the TAs can help you there. Yeah. Is it possible to get a TA? The links are not visualizing. Yeah, if anybody can help Liam to visualize the links. If it doesn't work, Liam, I am happy to stay behind after the session and then working with you on that too. Okay, so the next part, I'm going to talk about a specific example that was of interest to us and how we were using the FocalPointPlasticity plugin for that. But the goal for some of this is again extending on what we just talked about, that these focal point plasticity parameters can be changed in the steppable file at a particular point in time, at a particular, like, in between particular regions, so that you can assign these asymmetries. And that is what we were exploiting to sort of look at example of somatogenesis. So in our case, what we knew is that somatogenesis resulted in the creation of periodic breaks in the mesodermal tissue, in the presomatic mesoderm. And we wanted to check if application of mechanical forces, which results in a sort of like a bent and folded tissue shape, can lead to periodic breaks in the tissue. And what could be some of the parameters that could, periodically break a tissue. And the other feature that that was of interest to us was that this sort of like breaking of the tissue occurred in a wave. So if you looked at, for example, the interior part of the tissue versus the posterior part, you can see that there are more of these rounded blocks present on the anterior side first, and then you can start to see the structures forming in the posterior, but not yet. So this is an example where the properties like change spatially from the anterior to posterior. And so we wanted to also recapture this aspect of sequentiality, that can we periodically break down a tissue into discrete chunks if there is some sort of wave-like / anterior posterior activation in which cell properties change sequentially. And so that's what I'm going to talk to you about today. So if you want, the codes for this module are under 7_3_3. And feel free to download the code. So I'm not going to walk through every detail of the code because it's a little involved. But what I'm going to share is how we can use some of the compute cell properties to achieve these two things that I talked to you about. All right. So what is my goal here? My goal is sort of being able to build up an epithelium, but now I want properties to rather change more in a sequential anterior-posterior fashion. And so, for example, if my interior, or let's say the left-hand side of my simulation, is where I want properties and features to change first, followed by the posterior, how would I achieve that? And then once I see this particular simulation in which my cell properties or behaviors have changed, I'm going to sort of break down my focal point plasticity links to get these periodic breaks in the tissue. So the goal for the next couple of modules is the following. So we're going to start, start slowly changing FPP properties from the left to the right, so that the cells are going to start pulling on each other from the left. And we're going to measure how strong the forces are between cells from the left, all over actually, but we're going to estimate how strongly the cells are pulling on each other, where the maximum stresses / tensions are. And then we're going to use that criteria to sort of break down this tissue. And so one of the properties that I ended up using, which might be useful to you if you want to implement things in a wave, is one of the features using dictionaries. So I have a keyword in here called constrict, which is what I was using to monitor where my wave is at a particular time. So what I check is I have a parameter called wave speed. And every time my wave speed parameter is reached as a function of particular Monte Carlo steps, I activate my cells sequentially. The cells which are activated can now start pulling on their neighbors, whereas cells which do not have this label of constrict equal to 1, do not pull each other. Their FPP properties are symmetrical and they stay the same. So if you are looking at module 7_3_7, these lines are between 74 and 80, what I'm showing here. And so this is allowing me to sort of have an attribute of activating these links at a particular time point. And then I can choose which cells in a simulation from left to right get activated by this, these wave speed. So then the next feature, what I'm going to do is exactly what I said. So now the cells which I know have this constrict 1 attribute as 1, or basically it has been activated by that passing wave. If that condition is met, it can now change the FPP properties in those regions, and I'm slowly trying to change my lambda distance in this case. So I'm slowly increasing the lambda distance in between the cells. And so now what you're going to see is, as a function of this parameter, which I'm calling wave speed, you're going to start seeing that cells, as they pass from the left to the right, they're going to start like clumping together as they get activated by this FPP link and property. So if you just run the simulation module _7_3_3, you should be able to see this. And if you also want to try some of these parameters, so in line number 70, there's this parameter called wave speed. You can try changing it to 100, or you could try increasing it to 600, and you should be able to see the differences, the rate basically at which the cells are able to pull on each other. And this was the and then I'm writing down the code lines if you want to read this part for yourself. It's in step function. So now that I'm able to produce this, where I can sequentially sort of activate this over time and get the cells to pull on each other, I want to sort of think about breaking this tissue down. And I'm going to use, again, 2 features of the FPP property here again. And so the 2 properties are basically link deletion. So along with the link creation module that we went through, where we can pick out, say, neighboring cells, and we can form links between them, we can also break links between them at any point. And FPP plugin allows us to do that. And how do I break the link? When do I break the link? I can additionally use the features of what I see from the simulation to make that call. For example, as my cells get pulled together, what you'll see is that the cells which are at the periphery of where the wave has passed through, they have a little bit more wriggle room to sort of, change their shape to match this target constraint that I've applied to them by increasing lambda. But cells which are in the middle right now, they are sort of stuck because they really do want to change their target distance to match as that lambda property for them is increased. But because they're stuck in the middle, they don't have a lot of room to move. So now if I try to calculate the level of force that exists in between these links as a function of the target distance and the target length that they're able to achieve, what I'll find is that the cells in the middle are the ones that are most stressed out because they can't match that. And if I now provide a threshold that says that once you have crossed this threshold and you're way too stressed out, just break that link, this will allow me to create these, this will allow me to sort of break down this tissue into these units. And so the next part of this module, if you now are able to get a simulation in which just cells are pulling on each other, starting sequentially from the left to right, you can now download 7_3_4. And what you should be able to see is that I have additionally incorporated the condition where I'm trying to measure the cell tension, in this case, as a function of the lambda distance and as a function of the target distance. And I say that if there is a threshold, in this case, I've given it some number. If the threshold has been reached, then you start, and so the cells are way too stressed out. Now, you can try, the simulation should put them in this additional list. And then those are the links that will be deleted. And if you go to 7_3_4, these lines are under these are under 96 and 100. And the actual deletion takes place. So right now I'm just appending it to a list, but then I under lines 104 to 109 is where I am deleting those links. So if you run the simulation, now you should be able to see, now you should be able to break down the tissue. And I'll tell you upfront that there are two interesting things of this simulation, which was very cool. One was that wave speed that I talked to you about, like how fast you're basically pulling on each other. And then the other one is that rate at which you're increasing that lambda. And what we found out was that depending on how fast we are able to activate the cells in a wave versus how fast these cells are actually, pulling on each other, that allowed for different morphologies of breakdown. Basically, how periodic you were or how non-periodic you were depended on those parameters. And so if these are features that you want to think about, when to break the links, those are parameters you can play with. For example, like I said, line number 70 is where you have the wave speed parameter. You could try changing it. You could change the rate at which you're increasing lambda. That's in line number 93. You could even try changing you can even try changing the tension, the cutoff, which I'm picking to break the tissue. In this case, I have arbitrarily set it to 400, but you can set it to a higher or lower value and see how periodic versus non-periodic structures you get. And then, like I said, once we have these links appended for deletion, we'll be using delete FPP link to break that down. And that's in line number 104 to 109. And so what we can see over time is you end up getting these structures that have that are incorporating, that are breaking down And the question is, how can we make more, how can we achieve more periodicity versus how can we achieve, a particular feature of the tissue using these parameters? So are people able to get this to run? Even if periodic breaking of tissue is not what you're interested in, I'm hoping that the concepts of using dictionary attributes might be useful to people who want to use it, who want to have like a wave-like feature across cells, and hopefully the link deletion and the creation help you sort of being able to manipulate these FPP properties on the go. So let me know. Let me know if people are able to produce these breaks. Susan saying the physics of the situation might guide the wave frequency and breakage or Rayleigh instability. One would have to know the viscosity of the tissue and temperature. Absolutely, yeah. So we for our case, we had some idea, but a lot of the features we had were not experimentally testable. But yeah, absolutely, it would be cool to know the viscosity and the temperature. Oh, well, Vikram, that's great. So you're able to get these periodic breaks in a structure. And if you're able to do this, I would suggest changing some of these parameters now to see if how these breaks actually change. The advantages, if you have a large simulation, it's actually better because these are, like you saw that I have some fixed boundary cells on either side. And so some of the boundary effects come into play. But if you have a large enough simulation, then you could actually get more structures in the middle, which are more uniform than the ones you're seeing at the two edges. So the final step for us was going to be this 7_3_5. So the goal was, can we sort of visualize where we have the breaks and how uniform or periodic these structures are? So the goal of this was sort of manipulating either the tension threshold, the wave speed, and trying to see what kind of like structures emerge. And if you can add additionally a position for these breaks, or you can visualize where these breaks happen. So the code for this should be under 7_3_5. And what I have additionally done is added a plot to visualize the positions of all the breaks. And you can see how this graph changes as a function of all the internal parameters of the code. So this is what I have for you today. I'm going to stay back, and then if you have any more questions, and if you have, things that you have tried and you'd like to talk about more, but yeah, that was my goal for the module, to sort of show some of these FPP parameters and properties. And like I said, here we have just used it for sort of between compartments to just change the shape of the overall tissue, but you can use it for elongation just by putting links in between these compartments and changing the lambda distance between them, and that will sort of lead to a very elongated cells. Yeah. And if you have thoughts on this, please fill out the questionnaire. I'm happy to also help out if there are any additional questions. And thank you so much for your attention.
All right, great. Thank you, Priyom. So if everybody could please take a minute to fill out our last questionnaire of the workshop, presumably.
I had a question
That
for
would be
Priyam.
great. And then, yeah, we'll have some time here for questions for Priyom. Avishai, go ahead.
Yeah, it was a nice lecture, Priyam. So I was just wondering if it is possible, or if you have calculated the stress on the apical layer.
So,
The
sorry, Could you say that again? Sorry, I couldn't hear
So
you.
is it possible to measure the stress on the apical layer as they kind of like cluster and deform using FP links?
Right. So, yeah, it's definitely possible. So right now, the stress slash tension we have measured is at the apical level. Only, but I think you should be able to have a better measurement in terms of maybe like the cell change or shape change. And I don't know if you can do it at a more like pixel level calculation. So here the stress that we have defined is basically the derivative of the focal point energy function. And so that's why it's proportional to the lambda and the target distance. But obviously, it does not take into account that the apical surface has actually like rounded up or changed. So I think it's possible to have a more generalized definition of the stress of the entire cell. Yeah.
Okay. Yes. Thank you.
So maybe I can have a question. Abhishek, the hand you are raising is left over, right? I guess so.
Please,
Okay.
please go ahead and ask your question.
Yeah, so the stress, the stretch of the tissue that you were talking about, so the pressure, let's put it like this is due to proliferation of the cells, right? But this proliferation is not modeled. Would it be possible to put it into the model to have some proliferation to get it really growing?
Right. So in this case, the stress is not coming from proliferation. In fact, like there are, for example, transcription factors that can like, promote actin myosin density that can downstream lead to this, localized forces that you see in the epithelium. I know in the Drosophila embryo, there are like a cascade of, GPCR proteins that get turned on before these functions happen. But absolutely, proliferation, if you want to additionally model proliferation here, you can absolutely do it. And I think from module 6.1, what Julio showed, there was a simulation from a previous graduate student in the group, Dr. Abbas, who had actually done something like this, where along with the folding properties, he had incorporated proliferation. And that might be closer to the buckling kind of scenario that you guys were talking about too. So it's absolutely possible. The things that you'd have to pay attention during then is once you have a new cell created, how do you break the old FPP links that existed between cells and reform new ones? That is something that you'll have to decide. And how do you sort of want to like modify these new properties? But yeah, absolutely, proliferation can be incorporated. There is the usually in CompuCell 3D, there's this mitosis steppable. And there are other ways you can do it too, but the simplest way is basically you start increasing the target volumes of the cell compartments, and once they cross a particular threshold, they divide, and then you have a new cell type. So there are ways you can combine both.
And would it be possible to model stem cells, for example, by just cutting off the apical compartment of the cell, because then it's not differentiated, so we can just model the lateral and the basal part as stem cells, so to say, and then after a while, as soon as it's getting apical head, then it stops proliferation, and then it can break the links, or it can form the links actually, and those links then after a while have to be broken, so that proliferation is just limited to some cells, and then the linkage break that you showed us would be enough, right?
Yes, absolutely. You can definitely do that. So there are ways in which you can change the cell type. So you were specifically talking about the apical lateral, but I'm just
Yeah,
going to
it's
general.
just an example.
Right, right. Sorry. Right. So yeah, a general example case where you could start off with, say, a single cell type. You can call it a stem cell population. You can then have particular members differentiate. And usually what you do is if you loop over that cell type, and if you do cell dot type equal to self whatever, that particular cell's type could be changed, could be changed specifically. So you could definitely have a situation where once that cell type is reached, that cell no longer proliferates, because in your proliferation loop, you only implement proliferation for a specific cell type. So it's absolutely possible to have a specific cell type divide, to have proliferation for a specific cell type, and for focal point plasticity properties to exist between only specific cell type.
Yeah, and one thing I wanted to tell you, this is a great lecture. This was a great lecture, sorry. I'm impressed.
Thank you so much. I appreciate that.
Other questions or comments for Priyom? We're very grateful to her for coming back from Duke to give us this lecture.
I think there was a question from Avishek about the linear stress profile. So in this case, the energy function we chose was just linear in proportional to the length. But James can correct me if I'm wrong. There are ways that you can actually apply nonlinear forces in focal point plasticity as well. I do not know biology, like realistically, which one would be more true for your tissue. But yes, there are ways that you can change the linear relationship to a nonlinear relationship. This should be there in the manual somewhere. If you do not find it, let me know, and I can search for it.
Thank you. I was actually curious because in most of tissue mechanics models, like the stress strain profile is mostly nonlinear. So there are kind of like strain energy density functions specifically designed for several tissue-like behavior or contraction behaviors. So I was a little curious about that.
Yeah, so you can implement any of those that you choose.
Yeah, I see. Okay. So it will depend on the type I choose and how it is implemented.
It's not pre-implemented, though, so you will have to design. You would have to draw the equivalent springs and do it. The thing that you don't have is the equivalent of a dash pot. In other words, if you want the equilibrium length to change in time because of the tension, then you have to put that in by hand. You would write a little ODE model, which you might want to implement in antimony, for example, and have that control the target length of the spring.
I see. Okay.
But you can, if the force is or the energy is a function of the target length and the current length, you can make that linear, quadratic, any functional form you like. If you want it to be dependent on the history or on the derivative of the strain rate, for example, then you have to do that by hand. But you certainly can do it, and people have done it in CompuCell. It's a little bit more work to set up, though.
Okay, thank you.
And I think, just to add to James's point, going back to the previous lecture, so yeah, having a model loaded from SBML antimony, you were talking about calcium as well. So that could be specifically an external field, which has its own dynamics. And then you could combine the rate of increase or change to the FPP link properties as well. So that could control how fast your cells grow or change depending on external signals.
Yeah, I think it would be very interesting to implement some of those and create some demonstrations and material around them. I mean, of course, you might be publishable research as well, but I think it would be nice to have more examples of those kinds of applications in CompuCell. Even the solids application where you build a solid material, you pull on it and you plot the stress strain diagram. I've written simulations like that over the years, but I've actually never put them into the CompuCell package. Again, those could be good for people who are looking for hackathon projects and who don't have something in mind. That kind of creation of demonstrations can be really useful. And also opportunity to get your name in the CompuCell release if they're put into the package and just distributed.
I guess an interesting paper related to that could be Gilberto's Hydra simulations, where you were actually able to measure out some of the viscoelastic properties in CompuCell. I'm thinking about the 2000-ish paper.
That was an old paper, but yes, that was a nice example of what you can do with that kind of model.